Hello everyone and welcome back to Biochemistry BCH 302 for the College of Natural Health Sciences Bermuda. My name is Dr. Delcina Bean Burrows and this is lecture number 11. Today's lecture objectives are that at the end of this lecture you will understand what nucleic acid is and be familiar with the basic structure of nucleic acid. Our lecture discussion begins today with the DNA structure. Nucleic acids are biopolymers that are crucial to all known forms of life. They are made up of monomers, which are nucleotides made of three components, a 5-carbon sugar, a phosphate group, and a nitrogenous base. When the sugar is a simple ribose, the polymer is RNA, which stands for ribonucleic acid, and when the sugar is acquired from ribose as deoxyribose, the polymer is DNA, deoxyribonucleic acid. They are the most essential of all biomolecules and are found in all living things, where they create, encode, and then store information in the nucleus of the cells of every living organism. They also function to transmit and express the stored information inside and outside the cell nucleus to the inner processes of the cell and, finally, to the next generation of each living organism. The encoded information is stored and transmitted through the nucleic acid sequence, which provides the ladder step arrangement of nucleotides inside the molecules of RNA and DNA. RNA structure. Strings of nucleotides are fused to form helical backbone, normally 1 for RNA and 2 for DNA, and arranged into chains of base pairs carefully chosen from the 5 primary or canonical nucleobases: thymine, adenine, cytosine, guanine, and uracil. Thymine are found only in DNA and uracil only in RNA. With the aid of amino acids and the process of protein synthesis, the particular sequencing in DNA of these nucleobase pairs facilitates the storage and transmission of coded instructions as genes. In RNA, base pair sequencing aids the creation of new proteins that determine the frames, parts, and most chemical processes in all living organisms. Nucleic acid, which is a chemical compound that occurs naturally, can be broken down to yield phosphoric acid, sugars, and a mixture of organic bases, purines, and pyrimidines. They are the chief information-carrying molecules of the cell, and by guiding the process of protein synthesis, they determine the inherited characteristics of every living organism. The two main classes of nucleic acids are deoxyribonucleic acid, which is DNA, and ribonucleic acid, which is RNA. DNA can be defined as the master blueprint for life and constitutes the genetic material in all living organisms and most viruses. RNA is the genetic material of certain viruses, but it can also be found in all living cells, where it plays a key role in processes like the creation of proteins. Now, in terms of the basic structure, nucleic acids are polynucleotides, meaning that they are long chain-like molecules made up of a series of almost identical building blocks referred to as nucleotides. Each nucleotide contains a nitrogen-containing aromatic base attached to a pentose, 5-carbon, sugar, which is in turn attached to a phosphate group. Each nucleic acid has 4 of 5 possible nitrogen-containing bases. These are adenine A, guanine G, cytosine C, thymine P, and uracil U. A and G are grouped as purines, while C, T, and U are jointly termed pyrimidines. All nucleic acids have the bases A, C, and G. However, T is found only in DNA with U, while U is found in RNA. The pentose sugar in DNA is different from the sugar in RNA because of the absence of a hydroxyl group on the 2 carbon of the sugar ring. Without an attached phosphate group, the sugar attached to one of the bases is referred to as a nucleoside. The phosphate group joins consecutive sugar residues by connecting the 5-hydroxyl group on one sugar to the 3-hydroxyl group of the next sugar in the chain. These nucleoside linkages are referred to as phosphodiester bonds and are the same in RNA and DNA. Nucleotides are synthesized from already existing precursors in the cell. The ribose phosphate section of both purine and pyrimidine nucleotides is synthesized from glucose through the pentose phosphate pathway. The 6-atom pyrimidine ring is first synthesized and then attached to the ribose phosphate. The 2 rings in purines are synthesized while connected to the ribose phosphate during the assembly of adenine or guanine nucleosides. In these 2 cases, the final product is a nucleotide carrying a phosphate attached to the 5 carbon on the sugar. Lastly, a specific enzyme referred to as a kinase adds on 2 phosphate groups using adenosine triphosphate ATP as the phosphate donor to create ribonucleoside triphosphate, the direct precursor of RNA. When it comes to DNA, the 2-hydroxyl group is removed from the ribonucleoside diphosphate to create deoxyribonucleoside diphosphate. An extra phosphate group from ATP is then added by another kinase to create a deoxyribonucleoside triphosphate, the direct precursor of DNA. In the process of cell metabolism, RNA is continually being formed and broken down. The purine and pyrimidine residues are reused by numerous salvage pathways to form more genetic material. Purine is salvaged in the form of the corresponding nucleotide, while pyrimidine is salvaged as the nucleoside. Deoxyribonucleic acid. This is a polymer of the 4 nucleotides A, C, G, and T, which are joined through a backbone of alternating phosphate and deoxyribose sugar residues. These nitrogen-containing bases occur in complementary pairs as designated by their ability to form hydrogen bonds between them. A always pairs with T through 2 hydrogen bonds, and G always pairs with C through 3 hydrogen bonds. The spans of A, T, and G, C hydrogen bonded pairs are almost identical, allowing them to bridge sugar phosphate chains uniformly. Its structure and the molecule's chemical stability makes DNA the perfect genetic material. The bonding between complementary bases also gives a mechanism for the reproduction of DNA and the transmission of genetic information. In 1953, James D. Watson and Francis H.C. Crick offered a three-dimensional structure for DNA based on low-resolution X-ray crystallographic data and on Erwin Schrödinger's observation that in naturally occurring DNA, the quantity of T equals the quantity of A, and the quantity of G is equal to the quantity of C. Watson and Crick advanced that two strands of polynucleotides curl around each other, forming a double helix. The two strands, nonetheless identical, run in opposite directions as designated by the orientation of the 5 to 3 phosphodiester bond. The sugar phosphate chains run alongside the exterior of the helix, and the bases lie on their interior, where they are connected to complementary bases on the other strand by way of the hydrogen bonds. The double helical structure of a normal DNA takes a right-handed form referred to as the B helix. The helix makes one whole turn at around every 10 base pairs. B DNA has two principal grooves, a wide major groove and a narrow minor groove. Numerous proteins interact in the space of the major groove, where they make sequence-specific contacts with the bases. A few proteins are also known to make contacts through the minor groove. Quite a lot of structural variants of DNA are known. In aDNA, which forms under circumstances of high salt concentration and minimal water, the base pairs are slanted and displaced toward the minor groove. Left-handed zDNA forms in strands that have sequences with alternating purines and pyrimidines. DNA can form triple helices when two strands that have runs of pyrimidines interact with a third strand that has a run of purines. bDNA is usually portrayed as a smooth helix. However, certain sequences of bases can alter the otherwise regular structure. Most of these alternative DNA structures have only been categorized in the laboratory, and their cellular implication is unknown. Now, in terms of biological structures, we know that naturally occurring DNA molecules can either be circular or linear. The genomes of single-celled bacteria and archaea, the prokaryotes, are as well as the genomes of mitochondria and chloroplast are circular molecules, plus a number of bacteria and archaea have smaller circular DNA molecules referred to as plasmids that usually have only a few genes. A lot of plasmids are readily transferred from one cell to another. For a classic bacterium, the genome that encodes the organism's genes is a single contiguous circular molecule that has 500,000 to 5 million base pairs. The genomes of most eukaryotes and some prokaryotes have linear DNA molecules referred to as chromosomes. In all cells, DNA exists as a protein-coded complex referred to as chromatin. In prokaryotes, the loose coat of proteins on the DNA helps to shield the negative charge of the phosphodiester backbone. Chromatin also has proteins that regulate gene expression and the distinctive shapes of chromosomes. In eukaryotes, a segment of DNA between 140 and 200 base pairs curls around a distinct set of 8 positively charged proteins, referred to as a histone, creating a sphere-shaped structure called the nucleosome. Extrahistones are enfolded by consecutive sections of DNA, creating a series of nucleosomes that look like beads on a string. Transcription and replication of DNA is more complex in eukaryotes because the nucleosome complexes have to be at least partly disassembled for the processes to progress efficiently. Most, most prokaryote viruses have linear genomes that are usually shorter and have only the genes needed for viral propagation. Bacterial viruses, referred to as bacteriophages, can contain both linear and circular forms of DNA. Now let's take a look at the biochemical properties. There is chemical modification. After the assembling of DNA molecule, it can be chemically modified, sometimes intentionally by distinctive enzymes referred to as DNA methyltransferases, and at times accidentally by oxidation, ionizing radiation, or the action of chemical carcinogens. DNA can also be cleaved and degraded by enzymes referred to as nucleases. There is also the biochemical property of denaturation. The double strands of the DNA helix are joined together by hydrogen bonding interactions between the complementary base pairs. Heating DNA in solution easily breaks these hydrogen bonds, which in turn allows the two strands to be separated. This process is referred to as denaturation or melting. It is possible for the two strands to reassociate after the solution cools, reforming the initial DNA duplex. This process is referred to as renaturation or hybridization. These processes form the basis of several crucial techniques that are used for manipulating DNA. With the use of hybridization, a single-stranded DNA molecule can capture complementary sequences from any source. Single strands from RNA can also reassociate. DNA and RNA single strands can create hybrid molecules that can, at times, be more stable than double-bonded DNA. These molecules form the basis of a technique that is used to purify and characterize messenger RNA molecules corresponding to single genes. Ultraviolet absorption. The process of DNA melting and reassociation can be monitored by measuring the absorption of ultraviolet light at a wavelength of 260 nanometers. For double-stranded DNAs, absorption is fairly weak, but when DNA is single-stranded, the unstacking of the bases leads to an enhancement of absorption referred to as hyperchromicity. Thus, the degree to which DNA is single-stranded or double-stranded can be determined by monitoring UV absorption. Methylation. There are three types of natural methylation that have been discovered in DNA. Cytosine can be altered either on the ring to form 5-methylcytosine or on the azocyclic amino group to form N4-methylcytosine. Adenine can be altered to form N6-methyladenine. N4-methylcytosine and N6-methyladenine are found only in bacteria and archaea, while 5-methylcytosine is widely distributed. Distinctive enzymes referred to as DNA methyltransferases are responsible for this methylation. They can identify certain sequences within the DNA molecule so that only a subsection of the bases is modified. Other methylations of the bases or of the deoxyribose are, at times, induced by carcinogens. These often cause mispairing of the bases during replication and have to be removed so that it does not become mutagenic. Natural methylation has several cellular functions. In bacteria and archaea, methylation forms a crucial part of the immune system because they protect DNA molecules from fragmentation by restricting endonucleosis, or I should say endonucleases. In certain organisms, methylation removes incorrect base sequences that were introduced during DNA replication. This is done by marking the parental strand with a methyl group, a cellular mechanism referred to as the mismatch repair system, which differentiates between the newly replicated strand, where the errors happen, and the correct sequence of the template strand. In advanced eukaryotes, 5-methylcytosine controls numerous cellular processes by preventing DNA transcription. Methylation is also thought to signal imprinting, a process whereby some genes inherited from one parent are selectively inactivated. In mammals, methylation happens selectively within the dinucleotide sequence CG, a rare sequence, seemingly because it has been lost by mutation. In several cancers, mutations are found in important genes at CG dinucleotides. Nucleases. These are enzymes that hydrolytically slice the phosphodiester backbone of DNA. Endonucleases slice in the middle of the chains, whereas axon nucleases work selectively by degrading from the end of the chain. Restriction endonucleases are a unique group that identify and slice particular sequences in DNA. Type 2 restrictions endonucleases always slice at or near their recognition sites, and they create small, well-defined fragments of DNA that help to typify genes and genomes and produce recombinant. Fragments of DNA that are produced by restriction endonucleases can be transferred from one organism to another. Mutation. Chemical modifications of DNA may lead to mutations in the genetic material. Anions like bisulfite can deaminate cytosine to create uracil, altering the genetic message by bringing about C to T transitions. Exposure to ultraviolet light can lead to adjacent pyrimidines to dimerize, while oxidative damage from free radicals or strong oxidizing agents can cause a range of lesions that are mutagenic if not repaired. Halogens like chlorine and bromine react directly with uracil, adenine, and guanine, leading to substituted bases that are often mutagenic. Likewise, nitrous acid reacts with primary amine groups, which in turn leads to changes in base pairing and mutation. A lot of chemical mutagens like chlorinated hydrocarbons and nitrates are toxic because of the production of halides and nitrous acid during their metabolism in the body. Supercoiling. Circular DNA molecules can assume quite a bit of different typologies. There is active supercoiling, which has to do with cleavage of one DNA strand, its curving on one or more turns around the complementary strand, and then the resealing of the molecule. Each complete rotation causes the introduction of one supercoiled turn in the DNA, a process that can carry on until the DNA is fully wound and folds on itself in a tight ball. However, reversal is possible. Special enzymes referred to as gyrases and topoisomerases catalyze the winding and slackening of supercoiled DNA. In the linear chromosomes of eukaryotes, the DNA is typically tightly inhibited at several points by proteins, allowing the intervening stretches to be supercoiled. This phenomena is partly responsible for the compaction of DNA that is essential so that it can fit within the confines of the cell. The DNA in just one human cell would have a stretch length of between 2 and 3 meters, but it is packed very tightly so that it can fit within a human cell and nucleus that is 10 micrometers in diameter. Ribonucleic acid. This is a single-stranded nucleic acid polymer of the 4 nucleotides A, C, G, and U linked by way of a backbone of alternating phosphate and ribose sugar residues. It is the first intermediate when it comes to transforming the information from DNA into proteins that are necessary for cell function. Some RNAs also play direct roles when it comes to cellular metabolism. RNA is formed by copying the base sequence of a section of double-stranded DNA referred to as a gene into a piece of single-stranded nucleic acid. This process, referred to as transcription, is catalyzed by an enzyme called RNA polymerase. Now, in terms of the chemical structure, RNA is sensitive to oxidizing agents like periodate that lead to the opening of the 3-terminal ribose ring. The 2-hydroxyl group on the ribose ring is one major cause of instability in RNA, as the presence of alkali causes rapid cleavage of the phosdiester bond that links the ribose and phosphate groups. However, this instability is not a noteworthy problem for the cell because RNA is continually being synthesized and degraded. In RNA, which is typically single-stranded, the bases pair with other bases within the same molecule, leading to complex 3-dimensional structures. Sometimes intermolecular RNA duplexes do form, but they form a right-handed A-type helix instead of the B-type DNA helix. Subject to the amount of salt present, 11 or 12 base pairs are found on each of the helix. Helices between RNA and DNA molecules also form, but these adapt to the A-type conformation and are more stable than RNA or DNA duplexes. Such hybrid duplexes are formed when RNA polymerase transcribes DNA into mRNA for protein synthesis and when reverse transcriptase copies a viral RNA genome like that of the human immune deficiency virus, or HIV. Single-stranded RNAs are flexible molecules that can form a range of structures by way of internal base pairing and additional non-base pair interactions. They can form hairpin loops like those found in transfer RNA, or tRNA, as well as longer range interactions that involve both the bases and phosphate residues of 2 or more nucleotides. This leads to compact 3-dimensional structures. Now let's take a look at the different types of RNA. Messenger RNA, or mRNA, delivers the information encoded in one or more genes from the DNA to the ribosome, a specific structure or organelle where the information is translated into a protein. In prokaryotes, messenger RNA has a precise transcribed copy of the original DNA sequence with a terminal 5-triphosphate group and a 3-hydroxyl residue. In eukaryotes, the mRNA molecules are more intricate. The 5-triphosphate residue is further esterified, creating a structure referred to as a cap. At the three ends, eukaryotic mRNAs usually have long runs of adenosine residues that are not encoded in the DNA but are added enzymatically after transcription. Eukaryotic mRNA molecules are typically made up of small segments of the original gene and are made by way of a process of cleavage and rejoining from an original precursor RNA molecule, which is an exact copy of the gene. Generally speaking, prokaryotic mRNAs are degraded very quickly, while the cap structure and the poly A tail of eukaryotic mRNAs significantly improve their stability. Ribosomal RNA or rRNA. rRNA molecules are the structural components of the ribosome. They form extensive secondary structures and have an active role in recognizing conserved portions of mRNAs and tRNAs. They also help with the catalysis of protein synthesis. In the prokaryote E. coli, 7 copies of the rRNA's gene synthesized to close to 15,000 ribosomes per cell. In eukaryotes, anywhere from 50 to 5,000 sets of rRNA genes and as many as 10 million ribosomes can be present in a single cell. In eukaryotes, these rRNA genes are looped out of the main chromosomal fibers and coalesce in the presence of proteins to create an organelle referred to as the nucleolus. The nucleolus is where the rRNA genes are transcribed and the early assembly of ribosomes takes place. Transfer RNA or tRNA. Transfer RNA transports individual amino acids into the ribosome for assembly into the growing polypeptide chain. The tRNA molecules have 70 to 80 nucleotides and fold into a distinctive cloverleaf structure. There are also specialized tRNAs for each of the 20 amino acids that are required for protein synthesis, and in most cases there are more than one tRNA for each amino acid. The nucleotide sequence is transformed into a protein sequence by translating each 3-base sequence, referred to as a codon, with a specific protein. The amino acids are loaded onto the tRNAs by specific enzymes referred to as an aminoacyl tRNA synthetases, typically with one synthetase for each amino acid. Ribosomes. Not all catalysis within the cell is carried out exclusively by proteins. Ribosomes are RNA molecules that are able to catalyze specific biochemical reactions similar to the activities of protein enzymes. Discovered in 1982, ribosomes have been able to demonstrate that RNA can be both a genetic material like DNA and a biological catalyst like protein enzymes. This discovery has also further contributed to the RNA world hypothesis that suggests that RNA may have been central in the evolution of the prebiotic self-replicating systems. The most popular actions of natural or vitro-evolved ribozymes are the cleavage or ligation of RNA and DNA and peptide bond formation. Within the ribosome and ribozymes act as part of the large subunit ribosomal RNA that link amino acids during protein synthesis. They also take part in a variety of RNA processing reactions. This includes RNA splicing, viral replication, and transfer RNA biosynthesis. Some examples of ribozymes are the hammerhead ribozyme, the VS ribozyme, leadzyme, and the hairpin ribozyme. Antisense RNAs. Most antisense RNAs are synthetically altered by products of RNA or DNA within latent therapeutic value. Naturally, antisense RNAs have sequences that are the complement of the normal coding sequences that are found in mRNAs, also referred to as sense RNAs. Like mRNAs, antisense RNAs are single-stranded, but they cannot be transferred into protein. Antisense RNAs can deactivate their complementary mRNA by forming a double-stranded structure that blocks the translation of the base sequence. Artificially introducing antisense RNAs into cells selectively deactivates genes by interfering with normal RNA metabolism. Many viruses use RNA as their genetic material. This is mostly predominant among eukaryotic viruses, but a few prokaryotic RNA viruses are also known to do that. A number of common examples include poliovirus, human immune deficiency virus, and influenza virus, all of which affect humans, and tobacco mosaic virus, which affects plants. For some viruses, the whole genetic material is encoded in a single RNA molecule, whereas in the segmented RNA viruses, numerous RNA molecules can be present. Many RNA viruses, like HIV, use a specific enzyme referred to as a reverse transcriptase that allows the replication of the virus by way of the DNA intermediate. In certain cases, this DNA intermediate becomes assimilated into the host chromosome during infection. When this is done, the virus exists in a dormant state and effectively escapes the host immune system. Other RNAs. There are other small RNA molecules with specialized functions present in cells. A group example is the small nuclear RNAs, which are involved in RNA splicing, and a host of other small RNAs that form part of the enzymes telomerase or ribonuclease P are part of the ribonucleoprotein particles. The RNA component of telomerase has a short sequence that functions as a template for the addition of small strings of oligonucleotides at the end of eukaryotic chromosomes. Other RNA molecules serve as guide RNA for editing, or they are complementary to small sections of rRNA and either direct the location of which methyl groups need to be added or mark U residues for conversion to the isomer pseudouridine. Now let's take a look at RNA processing. Cleavage is the first that we will consider. After synthesis by transcription, most RNA molecules are processed before getting to the final form. Lots of rRNA molecules are cleaved from much larger transcripts and can also be methylated or enzymatically modified. Plus, tRNAs are typically formed as longer precursor molecules that are cleaved by ribonucleic P to create the mature 5' and often have extra residues added to their 3' to form the sequence CCA. The hydroxyl group on the ribose ring of the terminal A of the 3' CCA sequence acts as the amino acid acceptor essential for the function of RNA in protein building. Splicing. In prokaryotes, the protein coding sequence inhabits one continuous linear segment of DNA, but in eukaryote genes, the coding sequences are habitually split in the genome. The sections of DNA or RNA coding for protein are referred to as exons, and the non-coding regions separating the exons are referred to as introns. After transcription, these coding sequences must be linked together before the mRNAs are able to function. The process removing the introns and the succeeding rejoining of the exons is referred to as RNA splicing. Each intron is removed in a separate series of reactions by a complex piece of enzymatic machinery referred to as spliceosome. This machinery is made up of a number of small nuclear ribonucleoprotein particles, snRNPs, that have small nuclear RNAs. And we have RNA editing. Some RNA molecules, especially those in protozoan mitochondria, go through extensive editing after their initial synthesis. During this editing process, residues are added or deleted by a post-transcriptional mechanism under the influence of guide RNAs. In some cases, close to 40% of the final RNA molecule can be acquired by way of this editing process, rather than being coded directly in the genome. Let's now take a look at nucleic acid metabolism. The three main processes of DNA metabolism are replication, repair, and recombination. These are carried out by specific machinery within the cell. DNA must be replicated correctly to guarantee the integrity of the genetic code. Errors that occur during replication or that are due to damage after replication must be repaired. Lastly, recombination between genomes is an essential mechanism that provides variation within a species and assists the repair of damaged DNA. Replication. This is a semi-conservative process in which the two strands are separated and new complementary strands are generated independently, producing two exact copies of the original DNA molecule. Therefore, each copy has one strand that is derived from the parent and one newly synthesized strand. Replication starts at a specific point on a chromosome referred to as an origin, continues in both directions along the strand, and ends at a specific point. When it comes to circular chromosomes, the end is reached automatically when the two extending chains meet, at which point specific proteins join the strands. DNA polymerases cannot begin replication at the end of a DNA strand. They can only prolong already existing oligonucleotide fragments referred to as primers. Hence, in linear chromosomes, special mechanisms begin and end DNA synthesis to avoid loss of information. The initiation of DNA synthesis is typically heralded by synthesis of a short RNA primer by a particular RNA polymerase referred to as primase. After DNA replication, the initiating primer RNAs are degraded. The two DNA strands are replicated in varying ways dictated by the direction of the phosphodiester bond. The leading strand is replicated consistently by adding individual nucleotides to the three end of the chain. The lagging strand is synthesized in an intermittent manner by laying down short RNA primers and then filling the gaps by DNA polymerase so that the bases are always added to the 5 to 3 direction. The short RNA fragments created during the copying of the lagging strands are degraded when they are no longer needed. The two newly synthesized DNA segments are linked by an enzyme referred to as DNA ligase. This way, replication can continue in both directions with two leading strands and two lagging strands ensuing outward from the origin. Enzymes of replication. DNA polymerase adds single nucleotides to the three end of either an RNA or a DNA molecule. In the prokaryote E. coli, there are three DNA polymerases. One is responsible for chromosome replication, while the other two are involved in the synthesis of DNA during damage repair. The DNA polymerases of eukaryotes are much more complicated than this. A host of other proteins are also crucial for replication. Proteins referred to as DNA helicases help to separate the two strands of DNA and single-stranded DNA, binding proteins, stabilizing them in the course of opening prior to being copied. The opening of the DNA helix causes significant strain in the form of supercoiling. This is subsequently relaxed by enzymes referred to as topoisomerases. A distinct RNA polymerase termed primase synthesizes the primers required at the origin to begin transcription, and DNA ligase seals the nicks formed between individual fragments. The ends of linear eukaryotic chromosomes are marked by special sequences referred to as telomeres, that are synthesized by a distinct DNA polymerase referred to as telomerase. This enzyme has an RNA component that plays the role of a template for the precise sequence found at the ends of chromosomes. Several copies of a short sequence in the telomerase-associated RNA are formed and added to the telomere ends. This prevents the shortening of the DNA chain that would otherwise happen during replication. Recombination. This is the key mechanism by which variation is introduced into populations. There are two types of recombination, general and site-specific. With general recombination, which is also referred to as homologous recombination, there are two to do with two DNA that has long stretches of similar base sequences. The DNA molecules are cut to create single strands. These afterwards enter the other duplex, where base pairing leads to a four-stranded DNA structure. The cruciform junction inside this structure is referred to as a Holliday junction. This was named after Robin Holliday, who proposed the original model for homologous recombination in 1964. The Holliday junction travels along the DNA duplex by unzipping one strand and restructuring the hydrogen bonds on the second strand. After this branch migration, the two duplexes can be nicked again, allowing them to separate. Lastly, the nicks are repaired by DNA ligase. The result is two DNA duplexes in which the section between the two nicks has been replaced. A key enzyme involved in recombination is RECA, which catalyzes the strand invasion process. RECA coats single-stranded DNA and aids its pairing with a double-stranded DNA molecule that has the same sequence. This in turn produces a loop structure. It should be noted that the initial product of general recombination between two regions of DNA that are similar but not identical is a heteroduplex. That is, a molecule inside which mismatched bases will be present at some points in the helix. Therefore, in the specialized recombination that happens during meiosis, one round of replication is needed before the mosaic chromosomes created by the recombination are properly matched. There are enzymes present in cells that specifically know and repair mismatches so that the initial products of recombination can occasionally be repaired before they are replicated. In cases like this, the final products of replication will not be true reciprocal events, but instead one of the original parental molecules will appear to have been preserved in the marginalization of the other. This is a process referred to as gene conversion. Recombination can also function to sometimes repair lesions in DNA. When one chromosome of a pair becomes irrevocably damaged, the information from the other chromosome can be copied and inserted by way of recombination to offer a correct replacement of the damaged section. Site-specific recombination comprises of very short specific sequences that are recognized by proteins. Long DNA sequences like viral genomes, drug resistance elements, and regulatory sequences can be removed, inserted, or inverted. Site-specific recombination is accountable for reshaping genomes. Now let's talk about repair. It is crucial that the integrity of DNA is maintained to ensure the accurate functioning of a cell throughout its lifetime and to make sure that the genetic information is correctly passed from one generation to the next. This goal is attained through repair processes that continually monitor the DNA for lesions and trigger appropriate repair enzymes. Critical lesions in DNA, like pyridamine dimers or gaps, can be repaired by way of recombination mechanisms, but there are several other repair mechanisms. One of such mechanisms is the mismatch repair. The system is regulated by the presence of a methyl group in the sequence GATC on the template strand. Similar mismatches, or I should say similar systems for mismatch repair, also function in eukaryotes. However, the template strand is not marked by methyl groups. As a matter of fact, lesions in the genes for human mismatch repair systems are identified to be responsible for many cancers. Loss of the mismatch repair system allows mutations to form quickly and ultimately affect the genes that aid in cell division. Because of this, cells divide in an uncontrolled manner and become cancerous. After replication, the most rampant form of damage to nucleic acids is one in which the normal A, C, G, and T bases are altered into chemically modified bases that generally vary significantly from their natural counterparts. The only exemptions are the deamination of cytosine to uracil and the deamination of 5-methylcytosine to thymine. In these instances, the product is a G to U or a G to T, mismatch. Specific enzymes referred to as DNA glycosylases can identify uracil in DNA or the thymine in a G to T mismatch and can selectively eliminate the base by cleaving the bond between the base and the deoxyribose sugar. Several of these enzymes are precise for the different chemically modified bases that may be present in DNA. Another way of repairing DNA lesions is by an excision repair pathway. Here, enzymes identify damage within DNA, most likely by detecting an altered conformation of DNA, and then nick the strand or either side of the lesion. This will allow a small single-stranded DNA to be excised. DNA polymerase and DNA ligase then go on to repair the single-stranded gap. Now let's discuss RNA metabolism. RNA provides the link between the genetic information encoded in DNA and the actual activities of the cell. Some RNA molecules, like the rRNAs and the snRNAs, become part of complex ribonucleoprotein structures that within specific roles of the cell. Others, like tRNAs, play crucial roles in protein synthesis, whereas mRNAs direct the synthesis of proteins by the ribosome. Three distinct stages of RNA metabolism occur. The first is that the selected segments of the genome are copied by transcription to create the precursor RNAs. Secondly, these precursors are processed to become functionally mature RNAs that are ready for use. Thirdly, the RNAs are degraded after use and the bases are recycled. Therefore, transcription is the process through which a specific segment of DNA is copied into a specific RNA that encodes a single protein or plays a structural or catalytic role. Translation is the decoding of the information within messenger RNA molecules that takes place on a specific structure referred to as a ribosome. Okay, so we have reached the end of today's lecture, and for assignment number 11, I'd like for you to provide a definition for the following: DNA, RNA, list the different types of RNA, describe the process of RNA metabolism, and discuss the importance of DNA integrity. Now, of course, this assignment is due this coming Sunday, no later than 11:55 p.m. If you have difficulty with this assignment, please send me an email or shoot me a text message so that we can set up a time to discuss whatever you need to discuss referring to this assignment. And of course, my office hours are on Thursdays from 4 to 6 p.m. And with that, I look forward to receiving your assignments, and I will see you in the next and final lecture for this course.